that is transfer RNA. So transfer RNA genes, so the transfer RNAs are important for like transporting the amino acids at the site of ribosome, at the site of protein synthesis, okay, in ribosome where the polypeptide chain is synthesized. And other bacteria like Borrelia burgdorferi, so it is a linear chromosome like eukaryote. So see, you can see like both of them are bacteria, but one is having circular DNA and other is having linear chromosome. So that means like it varies, the genetic material varies from organism to organism. So this is the prokaryotic cell. Okay, so here if you can see the chromosomal DNA, it is simply dispersed in the cytoplasm. And apart from this chromosomal DNA, plasmid DNA, which is extra chromosomal DNA, is also present in this bacterial cells. So besides chromosomal DNA, many bacteria, they have some extra chromosomal genetic materials, which are small circular and closed DNA molecules called plasmids. So these plasmids, they can replicate themselves and they will have some markers. Okay, so therefore scientists exploited these plasmids and they started using plasmids as the vectors. So for transferring of genes. So that comes under biotechnology, that is recombinant DNA technology. And so these plasmids are utilized in biotechnology. And they generally remain floated in the cytoplasm and bear different genes based on which they have been studied. And different types of plasmids are there. So these extra chromosomal DNAs are necessary for bacterial cells, but they are not essential. That means without normal DNA, that is genetic material, they cannot survive. But without plasmids also they can survive. That means, but these plasmids help bacteria in different processes. Like what are the different types of plasmids? F plasmids, R plasmids, virulent plasmids, metabolic plasmids, etc. So F plasmids are involved in the mostly fertilization. So like for reproduction. Okay, so in that we have F plus, F minus plasmids, like R plasmids are there. Virulent plasmids are involved in virulence. Virulence is nothing but the ability to cause infection. So in that process, and some other metabolisms also they play important role, like for synthesis of antibiotics, etc. Then virus genetic material, so it can be DNA or RNA, so which adopts different structures here. So usually it is circular and packed inside a virus particle. Then eukaryotic genetic material, so it is the, we call them as genomic DNA. So the information of all the genetic material which is present in the body, no, all the cells, okay, that is called as genome. So this genomic DNA is enclosed in a organelle which is called nucleus. And within this DNA is organized in the form of genes, which are hereditary units. So these genes are present in the chromosomes. Okay, so each chromosome can have some genes. So like that, different chromosomes can have different genes. Okay, so this, how these chromosomes are formed? By chromatin network. So what is a chromosome? So chromosome, it was first revealed by Walter Fleming. He studied chromosome in the 1880s. Okay, so during cell division, when the cells are dividing, this genetic material, whatever the material is there, that will organize itself into a thread-like structure called chromosomes. So chromo means color, zomes means which are having the color. So how we can actually, if we see under microscope, we cannot identify like DNA, RNA or anything by naked eyes. So we have to stain them or we have to give some fluorescence or like that. Then only we can easily identify. So in the same way, when the scientists have studied, so they used some basic dyes and stains so that they could identify the chromosomes. So the term chromosome was coined by Walder in 1888. So the term, as I told you, chrome means color. Soma means body, that is colored bodies. And they have DNA and proteins. So DNA and protein together called as chromatin. Okay, this chromatin, when it is tightly packed, it will form chromosome. So these chromosomes consist of many genes. And other than genes, there are regulatory elements, nucleotide sequences. So gene is the one which codes for the mRNA and then which that ultimately codes for the protein. But the regulatory elements are again the small segments of nucleotides, but they don't code for any proteins. They are simply for like signals. They act as a signals, like for initiation of some process like replication, transcription, translation, or to stop something. Like for example, if we have protein synthesis, so to stop translation, we have stop codons, right? So the stop codons, they are like a regulatory elements. Okay, so they are also present on the chromosomes. And chromosomes also contain DNA bound proteins. These proteins are histones. So what is the role of these proteins? They will help in the packing of DNA and control its functions. And the number of chromosomes varies from organism to organism. So in the last class, I think I have given you I have shown you the table. Like each organism is having different number of chromosomes. Then Benden and Boveri, they have reported the number of chromosomes in each species is constant. That means suppose for human beings have some number of chromosomes, whereas monkeys will have another number. Plant will have another number. Okay, so that means this number of chromosomes varies from one organism to another organism. But within human beings, it will be constant. The same number of chromosomes are seen in all human beings. If they are more in number or less in number, then that causes some diseases. Okay, so this concept was given by Benden and Boveri. And then Sutton and Boveri in 1902, they suggested that chromosomes are physical structures which are acting as the messengers of heredity. So that means they are nothing but they pass on the message of how a person should be or how an organism should be. Yeah, so here also you can see the number of chromosomes. Like Arabidopsis thaliana is a plant. It is having, this is 2n number that is diploid. So that means 5 chromosomes which are in 2 numbers, that is 2n is 10. Maize is having 20 chromosomes. Wheat, they are hexaploid. See, 6 sets. Okay, hexa means 6. So now 42 by 6 you have to divide. So that means 6 copies of chromosomes are present. Then fruit fly, Drosophila, which is having 8, that is diploid. That means 4 chromosomes in 2 sets. Earthworm is 36 chromosomes. Mouse, it is also diploid. So most of them are diploid. Only wheat is hexaploid. So mouse having 40, human 46, that is 23 pairs. Elephants 56, donkey 62, dog 78. So goldfish, it varies from 100 to 104. And tobacco, you can see this is tetraploid. That means 4 sets. And oats, they are having hexaploid. So if you see the plant crop species like wheat, oats, so they are having like hexaploid, right? So that means, see, you can see each species having different number of chromosomes. So these chromosomes are tightly coiled around basic histone proteins. And during interphase, DNA is not tightly coiled. It simply distributes in the cytoplasm. But it exists as a chromatin. Chromatin is nothing but the DNA and proteins. So both DNA and proteins are present, but they are not packed. And so to fit the length of the DNA is very long. If you like, I don't remember exact thing, but if you like add end to end of all the genes or all the like genetic material which is present in all the cells of our body, like if you take one human being, just remove genetic material from all the cells of the body, and you keep like you arrange the DNA as one after the other, so joining at the end. Okay, so one after other, then that will be of distance from earth to moon, some number of times. I don't remember exactly, 3 or 7 times it can, like the length is that much. Okay, so that much amount of DNA has to be packed in a very tiny nucleus, which is not even, which we cannot see with our naked eye. So how much condensation or packing it needs to be? So it will be tightly packed and condensed, and that will be put in the nucleus. Okay. So the shortest human chromosome, it consists of 4.6 into 10 power 7 base pairs of DNA, and its length is 14,000 micrometers. But when it got condensed, now the chromosome should be 2 micrometer. That means, see, 14,000 micrometer length DNA has to be packed into 2 micrometer small chromosome. So therefore, the packing should be 7,000 times like tighter. Okay, so the condensation should be around 7,000 times. So this is how efficiently it will pack. And it is packed in a stepwise, not simply randomly, but stepwise process is there. So this is called as hierarchies of chromosomal organization. So the first step is, the first level of packing is binding of DNA around protein. So here you can see the first image. See, this is the DNA, normal DNA which is distributed. Then these yellow colored ones are histone proteins. So there are 8 histone proteins, 2 sets of H2A, H2B, H3 and H4. So 4 proteins which are 2 copies. So all of them form 8 proteins. They form a core, and this DNA is wound around these histone proteins, and they form nucleosomes. This each histone along with the DNA, it forms the nucleosome. And the linkage is by linker DNAs, that is H1. This is also histone protein, but it is called linker DNA. So around 146 base pairs of DNA is wrapped around each nucleosome. This is called as nucleosome. In the second level, these nucleosomes are further condensed. Okay, they're tightly packed. You can see here the chromosomes are extended. Okay, then in the final step, they form scaffolds, loops or domains, and they tightly pack these nucleosomes together, and now they will form a structure called chromosome. Okay, and this is the chromosome, and this is the centromere where the 2 chromatids will join, and these chromatids have a long arm and short arm, and 2 sister chromatids, they join at the center by centromere. So this we can see in the mitosis. Okay. So this is how the packaging takes place. Now the structure of chromosome. A chromosome of mitotic metaphase consists of two symmetrical structures called chromatids, and each chromatid consists of single DNA molecule. Now both these chromatids are attached together by centromere, and during anaphase they get separated. Okay. And as I told you, like bead-like structures will be present. They are called nucleosomes, all these things. And this chromomere bearing the chromatin has an appearance of necklace. So if you see in a necklace, all the beads are arranged one after the other to a string. So in the same way, we can see like in a chromomere, which is having different nucleosomes. So here the image. So here the first one, where you can see they are like beads which are like which are arranged like a necklace to a string. Okay, this is how it looks like. Now what is centromere? So centromere in a chromosome contains specific DNA sequence with special proteins, and they will have a disc-shaped structures called kinetochore. Okay, and this kinetochore is like a cup or plate shaped. So where the constriction, that means a small bending or both the chromatids, they come together at the centromere. And the chromosomes contain only one centromere usually, and they are called as monocentric chromosomes. But some may have more than one also, but commonly they contain one centromere. So besides the primary constriction of centromere, sometimes they can have a secondary constriction also. Okay. And these constrictions are helpful in identifying particular chromosome in a set. And the secondary constrictions contains the genes which are coding for 5.8S, 18S, 28S ribosomal RNA. So if you remember when I discussed about the ribosomes, I discussed about the small subunit and large subunit. So each subunit is having different ribosomal RNAs. So the genes which are coding for that RNAs, they are located in this secondary constrictions. Sometimes they also can have some knob-like structures which are called satellites. So satellites usually having, satellite means a small sequence which will be sometimes repetitive, okay, repeating unit, that is called satellite. Now chromatin. So chromatin consists of DNA, RNA and protein, and proteins are two types, histones and non-histones. So DNA, so as we have discussed, like it is it codes for heredity, and it has a number of like total amount of DNA. So it is packed in the chromosome. So all this, so whatever we have discussed. So in addition, the amount of DNA per haploid cell, okay, or the number of kilobase pair per haploid cell is given as C value. So this is important. So we have a concept called C value paradox. So C value paradox is nothing but the C value varies from organism to organism. So if you normally we have diploid set of chromosomes, that is 2 copies. If only one, that is haploid set of chromosome is present, so the amount of DNA present is given as C value, and this differs from organism to organism. So, and this C value is constant for the species. That means in the diploid cell, we have 2C. That means 2 copies of the C value. So this is, I told you, like C value paradox. So what is the C value paradox means? So there are some like to the hypothesis and to the observation. So there are some differences. So that was found by the scientists. So that means I will explain you here. So one immediate feature of eukaryotic organisms highlights a specific anomaly that was detected early in molecular research. Even eukaryotic organisms appear 2 to 10 times as many as genes as prokaryotes, and they have many orders of magnitude more than DNA in the cell. So that means, so according to hypothesis, what the like scientists have thought is, as prokaryotes or primitive organisms, they will have less amount of DNA and less complex. But as the complexity of the organism increases, even the complexity of DNA also increases. So this is the hypothesis. But in actual, like we could see that sometimes bacteria is having more amount of DNA than higher organism and more complex also. So more amount of DNA per genome may be present. So this is called C-value paradox. That means the amount of DNA in the haploid cell of an organism is not related to its evolutionary complexity. So we cannot simply assume that as the organisms are evolved highly, they should have more DNA. No, it is not so. So this is termed as C-value paradox. So lower eukaryotes in general have less DNA, such as nematode. So in nematode, best example is Caenorhabditis elegans, but it has 20 times more DNA than E. coli. So you can assume E. coli. So that is Caenorhabditis elegans is having 20 times more DNA than the E. coli. So we assume that E. coli is more evolved than the Caenorhabditis elegans. And even the eukaryotes, lower eukaryotes, sometimes can have less DNA. Okay. And vertebrates has greater DNA content, that is 3 picograms, which is 700 times more than the E. coli. And salamander, which is having high DNA content of 84 picograms, whereas human being has having about only 3 picograms of DNA per haploid genome. See, salamander is having 84 picograms. So it is nowhere comparable. It is very high. That means the complexity is not related to the genetic material present. Okay, is it clear? This is C-value paradox. The next is histones. Histones are the proteins. Okay, histones are the basic proteins. They are basic in nature, and because they have basic amino acids. Okay, so if we see in the amino acids, there we have 20 amino acids. So in that we have acidic amino acids, basic amino acids, then aliphatic, aromatic amino acids, and we have different types of amino acids. So the basic amino acids like arginine and lysine are present in the histones. Therefore, they are basic in nature. And at physiological pH, they are cationic, and they can interact with the anionic nucleotides. So DNA is usually negatively charged. Histones are positively charged. Hence, they can interact. So they can form a highly condensed structure. And these histones are of 5 types: H1, H2A, H2B, H3, and H4, which are all similar and conserved. But H1 histone is usually act as a linker protein, whereas other 4, they have 2 copies and they will form a core. H1 is loosely bound to the DNA, and H1 is completely absent in the Saccharomyces cerevisiae, that is yeast. Then apart from histone proteins, we also have non-histone proteins. So in addition to histones, the chromosomes comprises of many different types of non-histone proteins, which are involved in the DNA replication and gene expression. Like in the replication, we have DNA binding proteins, helicases. So like that, we have many proteins which will come and bind to the DNA. And they have diversity and they are not conserved. And from tissue to tissue also, they may differ. Then we have two types of chromatin. They are euchromatin and heterochromatin. U means true. So what is euchromatin? So it is a lightly stained region in chromosome where it is stained with the basic dyes, and that is called euchromatin. So that means the chromatin is loosely packed. Hence we can stain them lightly. Okay, because the dye gets dispersed and the stain will be light. And the extent of chromatin condensation also vary during the life cycle of the cell. And it is important for regulation of gene expression because they are loosely packed. So the regulatory elements or regulatory proteins can easily access the DNA molecules or the content, and it can regulate. So in the interface, so whatever the chromatin which is decondensed, that is euchromatin, which is leading the gene transcription and DNA replication. Because only in S phase the replication takes place. Therefore, it should be accessible to all the proteins. Then heterochromatin. So the word heterochromatin was coined by Hades. So when he observed under the microscope. So it is highly condensed, okay, tightly packed, and therefore you cannot access, it is not accessible to proteins and we cannot regulate them. Okay, so about 10% of the interface chromatin is heterochromatin and highly condensed, and it is darkly stained because it is tightly packed. The stain will remain at the, it will be tightly associated with the chromatin. And it will have usually repetitive DNA, which is found in the centromere and telomeres. So telomeres are at the ends of the chromosomes, whereas centromere is at the center of the chromosome. So there you can see the heterochromatin. So heterochromatin is again of two types, constitutive, facultative. So the regions that remain condensed throughout the cell cycle are constitutive. Constitutive means continuous. Okay, so continuously they are condensed. Whereas the regions which change the condensation state, they are facultative. That means they are not strictly condensed all the time. So constitutive heterochromatin is seen mainly in the flanks of telomeres and centromere of chromosome in the distal arm and Y chromosome in mammals. Okay, and the constitutive chromatin consists of few genes, and it results in the transcriptional inactivation of nearby genes. That means it will have some genes and it will also inactivate the nearby genes which are present like in the proximity of the constitutive heterochromatin. And this phenomenon of gene silencing is called as position effect. Because they are staying near to the constitutive heterochromatin, they are getting inactive. So that is because of the position of that genes. And this constitutive heterochromatin also inhibits genetic recombination between homologous repetitive sequences. So usually there is a recombination process which takes place. That means one segment of the chromatin, it can get exchanged with the other segment. And here between non-sister chromatids. And this results in DNA duplications, deletions. And this constitutive heterochromatin usually it prevents the genetic recombination. Whereas facultative heterochromatin is specifically inactivated during the some phases of the life of the organism. So dosage compensation of X chromosome or X chromosome inactivation is an example of heterochromatin. So that means usually females, they will have two X chromosomes, but only one X chromosome will be active, other will be inactive. And this is like, that means if two X chromosomes are there, then we should have the double the amount of proteins which are present on the of the genes which are present on the X chromosome. But usually that is not the case. So they will be, one X chromosome will be inactive. Okay, So this is done by the heterochromatin. This is dosage compensation. So centromeres are the condensed regions which are present at the center, and they are helpful in the segregation of replicated chromosomes, that means separation during mitosis and meiosis, and they are darkly stained. They consist of spindle fibers and form a button-like structure called kinetochores, which have DNA and protein. And the DNA which is present in this region, kinetochore, is called cDNA, CEN DNA. So this is moved from one chromosome to another, and it has the ability to segregate. This cDNA is having 120 base pairs and consists of some subdomains like CD1, CD2, CD3. No need to remember all these things. Just remember that it is made up of DNA and protein. That's enough. So this is how it looks like. Okay, this is for just for your understanding. This is in Saccharomyces cerevisiae, that is yeast. So yeah, the next is telomeres. So telomeres are present at the region of DNA at the end of the linear chromosomes, at the ends, and this is to maintain the stability of chromosome. That means during replication, the edges may be lost sometime. So if that continues, so usually after every replication, we will lose some amount of genetic information. And these telomeres are mainly involved in the aging of the organism. So therefore, there is enzyme which is called telomerase, which will try to cope up the telomere length. It will maintain the telomere length. So McClinton, she recognized that this feature, and that if two chromosomes were broken in a cell, the ends were sticky and end of one could attach to the other. Okay. And however, she never observed the attachment of the broken end to the end of an unbroken chromosome. That means the end of chromosomes have unique features, so which are called as telomeres. So these telomeres are conserved throughout the vertebrates, and they are like caps to the chromosomes so that they protect the chromosome from nucleases and other destabilizing factors. Okay, so and it also prevents the fusion of ends of the chromosome. And this telomeric DNA consists of repeated sequences which are tandemly repeated. Tandemly means like it is repeated one after the other. Okay, that ATGC, then ATGC, like that. And human telomeres consist of the sequence which is TTA-GGG, which is repeated 500 to 5,000 times. Okay, and certain bacteria possess telomeres in the linear genetic material, which is two types. One type is hairpin telomere. The name itself, that means it looks like a hairpin. That means it is having a bend where the complementary strand is present. And the other type is called as invertron telomere. Here they overlap that means the overlap between the ends of the complementary DNA strands. That means inversion is possible. Then we have some giant chromosomes, which are special types of chromosomes. So they are large size, usually in salivary glands. There we can see this. They are called polytene or lamb brush chromosomes. Okay, they are larger in size. So polytene chromosomes was first observed by E.G. Balbiani in 1881 in salivary glands nucleus of Diptera. Okay, larva. Okay, and then this was first time reported in the insect Drosophila fruit fly by Theophilus Painter of University of Texas. And since they found this in the salivary gland of insects. They termed it as salivary gland chromosomes. So they have high DNA content. Sometimes they can also occur in Malpighian tubules, rectum, gut, foot pads, fat bodies, etc. And they are involved in that happens because of replication of chromosomal DNA several times, but there is no nuclear division. Okay, that means DNA is replicating, but the nucleus is not dividing. Okay, so therefore they align side by side, chromatin. So therefore this process is called endomitosis. That means within the same nuclear envelope, the replication of DNA is taking place. So these polytene chromosomes can be seen in interphase or prophase of the mitosis. They are 100 times thicker and they contains 1000 to 2000 chromosomes than chromosomes which are seen in other organisms. And they are tightly coiled regions which are present. They are called DNA fibers which are folded loosely. And when stained, they will appear dark bands. So, okay, we have like Q banding, all these different types of banding techniques are there. So here we can see a series of dark bands and interbands are clear zones. That means one dark band and then clear zone and another dark band and another clear zone. So like that it appears many number of bands, like 5000 bands are seen. So 85% of the DNA has dark bands and 15% is having interbands which are lightly stained. And so usually in cytogenetic studies, these are studied. And okay, so yeah. So this is how they look like, the polytene chromosomes. So they are very large in size. It is like a puff. Okay. These chromosome puffs are called as Balbiani rings because he has identified. So puffs may appear and disappear depending on the production of specific proteins. And another peculiarity is that it is paternal and maternal chromosomes remain associated side by side, which is called a somatic pairing. That means they are not separated. They are attached one after the other. The next is lamb brush chromosomes. This is because it appears to be a brush shaped. Okay, so usually we will have lambs. Okay, so in olden days we used to have lambs to steady, for the steady purpose. So there are small brushes which are used to clean these lambs. So these chromosomes appear in that shape. Therefore, they got lamb brush chromosomes. So this was first observed by Fleming in salamander oocytes and later described by Ruchter in 1892. They appeared like brushes for cleaning lambs. Hence, they got lamb brush chromosome name. So the structures are transitory in diplotin stage, usually of prophase 1 of meiosis. We can see this. And they are also seen in sepia, that is mollusca, echinodermata, and other species, insects, shark, amphibians, birds, reptiles, even in mammals like humans also they have been described. They have been seen in spermatocytes of some species. So that in the giant nucleus of Acetabularia and in some plants. They are smaller, the size will differ. Okay, so in invertebrates than vertebrates, and they have high DNA content, and usually we can see them in meiotic prophase, and these are helpful in construction of cytological maps of individual chromosomes. So this is how the lamb brush chromosomes looks like. So this is the pair lamb brush chromosomes. So this is a region of single chromosome. Okay, how it looks like. That means, see here, you can see it is like a brush. So small loops are attached. So therefore, as it appears like a brush, it got the name lamb brush chromosome. So this is the structure. They are exceptionally larger in size and present in bivalent forms. So two valent. So you can see here two loops, one loop at the top and other loop at the bottom. So that is bivalent. And they form due to the active synthesis of mRNA molecules for future use of the egg cells, because they are found in oocytes. And no synthesis of mRNA molecules is possible during the mitotic cell division. And we can see them under light microscope. And each loop consists of a constant, they are present at a constant position in each chromosome. And the loop has DNA which is unfolded from the chromosome and is transcriptionally active. And various transcriptional units with RNP, that is ribosomal proteins, okay? They are present in the loop of this DNA. And the majority of DNA remains highly condensed. That means only at the loop, the DNA is freely available, and other than the loops, the DNA which is present in the other regions, it is highly condensed. So the loops perform intense transcription of heterogeneous RNA. So that means heterogeneous RNA are the precursors of mRNA molecules for ribosomal and histone proteins. And the number of pairs of loops actually increases during meiosis, and it will become maximum at diplotin stage. And this may takes place for months and years as the oocytes build up the mRNA which is required for the egg cell formation. Okay, these are the various components and extra structures which we see in the chromosomes. Now we discuss about the genome complexity, how this genome is evolved and how complex they are. So 40 years ago, it was thought that the amount of DNA in the genome is correlated with the complexity of organism. That means smaller organism will have less complex DNA, whereas larger or highly evolved organism will have more complex DNA. But then, as we have already seen, like C-value paradox, so it is nothing like that. So this was like the graph, okay, which they have shown at that point of time. So it appeared like this. The prokaryote, like bacteria, is having less complex DNA than one-celled eukaryotes, then fungi, invertebrates, chordates, vertebrates, and then human beings. Human beings are having highly complex DNA. But in 1960s, scientists have started to look for the complexity of genome. So till that time, the genetic techniques were not evolved so far. Okay, now we have various high throughput techniques like next generation sequencing and many more techniques. So by using these techniques, actually scientists have started sequencing the genome, and then they could find some amazing facts. Okay, so larger genomes, they have repetitive sequences. So there are two types of sequences in our genome. One is repetitive sequence and other is unique sequence. Unique sequence is present only once, whereas repetitive sequences are having same sequence is repeated many number of times. And the amount of unique sequence is only few % of the total genome. And if you see only 5%, I don't remember exact percentage, but only 5% is a functional DNA, whereas other 95% of the genome is junk DNA. That means no proteins are, it is not coded for any protein or anything. So it is just present. It is a repetitive DNA. So that means even though we have millions of number of base pairs, only few, maybe 5% is functional. Okay. So now we see that genome size is not correlated to the complexity of the genome. So the range of DNA sizes varies from groups to, means varies from organism to organism. So mammals don't have any more DNA in their genome than most flowering plants. So it is similar to the flowering plant, same amount of DNA is present. And the genomes of most eukaryotes are larger and complex than those of prokaryotes. But the genes, the number of genes are almost similar. Okay, so that is what? That is till sequencing, the scientists couldn't find this fact. However, the genome size of many eukaryotes does not appear to be related to the genetic complexity. So the genome of salamanders and lilies contains more than 10 types of amount of DNA which is present in the human genome. So they are like usually in our thought that they are lower organisms, salamanders, and humans are highly evolved organisms, but they have a high amount of DNA than the humans. So this is the genome size. Now after sequencing. So this is Haemophilus influenza. It has high amount of uh, genomic DNA. Then Mycoplasma E. coli, which is bacteria. In fungi, we have yeast. In plants, Arabidopsis and lily. These were sequenced. Okay. And insects, Drosophila genome was sequenced. Then in amphibians, frog and salamander. Then birds, chicken, and mammals, human. So you can see they are represented in the logarithmic form. So here you have log scale. So these humans are between 10 power 9 to 10 power 10 in between somewhere. But salamander is around 10 power 11, even lily, right? So that means salamander and lily are having more amount of DNA when compared to human beings. So this apparent paradox. So this is called as a C-value paradox. So it is that the genome of eukaryotic cell consists of non-functional genes. So large amount of junk DNA is present. But still scientists are looking for the purpose of the junk DNA. So there is nothing like without any purpose. There may be some purpose for the junk DNA also, but so far it is not revealed. So the presence of large amount of non-coding sequences is a general property of genomes of complex eukaryotes. So human genome consists of 1 lakh genes, that is 25 times more than E. coli. Okay, so these are some of the facts. So how the structure of eukaryotic genes look like. So usually in prokaryotes, we don't see any introns and exons. So completely everything is they are arranged side by side and they can code for the proteins. But in eukaryotes, we have the coding sequence, which are called exons, which are interrupted by the non-coding sequences, which are called introns. So these introns, after transcription, they will be spliced. So this is the chromosomal DNA, you can see. So these introns are removed and all these exons are attached side by side to form a primary RNA transcript. And after primary RNA transcript, splicing takes place and these introns are removed. And finally, we get the mRNA without any introns. And once the introns are removed, so these introns are removed at intron-exon junctions. Okay. So once they are removed, only the mRNA is present with only exons, and then the translation takes place to give rise the protein. So introns have no cellular function. So therefore they will be removed. They became a part of gene during evolution, but they don't code for any proteins. Only exons will code for the proteins. Okay. Sometimes the recombination between introns of different genes, which results in new genes, okay, they can form a novel combinations of protein coding genes. So this type of hypothesis and research is still going on. We cannot conclude from this. Okay. So according to this hypothesis, introns played an important role in the initial protein coding genes of the ancient ancestors, but now they don't have any role. Okay. So that is the hypothesis. So maybe in future, after research, scientists may find some like purpose for the introns. So then we have some gene families and pseudogenes. So gene families means a group of genes which are the sequence is similar and they perform similar function. So another factor contributing to the larger size of eukaryotic genome is repetitive DNA, and most prokaryotic genes are represented only once, while eukaryotic genes are represented in multiple copies, which are called gene families. So they produce RNAs and proteins which are required in larger quantities. Like if you see rRNA and histones, they require in larger quantities. Therefore, these gene families are transcribed in different tissues and at different stages of the development. And like this amount will vary. It is not constant always. That means suppose during growth or during cell division, we require more proteins. So at that point of time, we require more amount of coding of such proteins. But when there is no cell division, then we don't require that protein. So at that point of time, the transcription cannot be seen. Okay. So example of gene families is alpha and beta subunits of hemoglobin. Both are encoded by the gene families of human genome. So there are two subunits. Only when these two subunits function together, then only they can perform their function, right? And this will vary, like whenever blood cell formation takes place, during that stage we require more amount of alpha and beta subunits, and during normal time we don't require, right? And even during the pregnancy, like embryonic stage or fetal stage, even in adult tissues, whenever after 120 days the blood cells will synthesize in the bone marrow, and during that stage then we need more expression of these genes. So this is nothing but the duplication of the original gene. That's it. So they together, as they are related, we call them as gene families. Okay. And sometimes we have non-functional gene copies which are called pseudogenes. Pseudo means false. So pseudogenes, that means they might have had some function during evolutionary process, but then due to mutation or something, they lost their function, but still they are present. So they are non-functional gene copies, the copies of the original genes, but because of mutation, they became non-functional. Okay. And this repetitive DNA sequences. So a substantial portion of the eukaryotic gene consists of highly repetitive non-coding DNA sequences. Sometimes we can have thousands of copies of repetitive sequences. So this was demonstrated by Britton and Cohne. Okay, this is by denaturation and renaturation process. So I'll just briefly discuss here. So what they have done is, like first they have isolated the complete genome, and they have like broken down the genome by using like various enzymes like restriction endonucleases, and they got small fragments. And then first they have separated the DNA strands. So that is by the process called denaturation. That means the separation of DNA strands is called as denaturation. And this may be due to pH change or addition of chemicals or by increase in temperature. So many factors can be used to perform this denaturation. Again, when the like the condition is restored, that means suppose first we have given the high temperature, then they get denatured, and if the temperature is lowered, again they get renatured. Okay, so like what they have done is first they have taken many copies of genome and they have denatured, and again they have restored the conditions so that again they'll get renatured. So during the process of renaturation, what they have found is some DNA molecules are renatured quickly and some DNA molecules they renatured slowly. So why this is because the repetitive DNA. See, suppose you have a segment called, I will just give X. So X is present in 100 copies. X has to fuse with Y. So you have 100 copies of Y. So then if you add these 100 copies of X and 100 copies of Y in a tube, so any X can fuse with any Y. So it will take very quickly. Okay, it cannot choose like which Y I have to fuse. It is not like that. So because they are complementary and there are more number of copies, any copy can go and fuse with the other copy of the Y segment. So therefore it became quickly. But for the unique DNA, as it is only single copy, it has to choose for the perfect complementary sequence with which it has to renature. So therefore it takes some time. So during this process, they could identify that there are repetitive DNA and unique DNA. Okay. Is like short sequences, like from 5 to 200 nucleotides that will be repeated one after the other. And this is seen in Drosophila, like ACA, ACAA, ACT. So this is repeated. Okay. And another is satellite DNA. So satellite DNA means they separate from the main band of the DNA. So they can repeat in millions of times in the genome. So 10 to 20% of the DNA consists of this type of DNA. So like this, we have different types of repetitive DNAs. And now, overall, what are the number of genes which are present in the eukaryotic cells? So suppose in bacterial genome, so most of the DNA codes for proteins, and the genome is 4.6 into 10 power 6 base pairs, and it has 4,288 genes, and 90% of the DNA is protein coding. But in the case of human beings, it has 3 into 10 power 9 base pairs, but only small amount of DNA is protein coding. Okay, many are repetitive pseudogenes or like this. So like this, if we can see here, E. coli, its genome size is 4.6 million base pairs, whereas Saccharomyces cerevisiae is 12 million base pairs. Caenorhabditis elegans 97 base pairs, million base pairs. Drosophila 180 million base pairs, and humans 3000 million base pairs. But see how much protein coding segment is present. E. coli 90% of the genome codes for protein, whereas human beings only 3% codes for the proteins. So that means it is not that how much amount of genome is present, but how much amount of genome is coding actually for the proteins. So this is all about the chromosomes, what is the organization, what are the different types of chromosomes, and what is C value paradox, and how much amount of like chromosomes and DNA are present in the various organisms. Okay, now we discuss briefly about cell cycle. So I'll stop sharing this. So this is the cell cycle. So it is the life of a cell. So what is cell cycle? Because it is a cyclic process, we call it as cell cycle. It is nothing but the like division. It is a cell division. Okay, how a cell divides into two. Okay, so it like follows some steps to divide. Okay, this is, we can call cell cycle, cell reproduction, or cell division. So cells must reproduce before they die. Okay, so and the cell life of a cell is called as a cell cycle. So in eukaryotes usually we have different phases, that is G1, S, G2 and M. So G1, G2 are gap phases, and S is synthetic phase, M is mitosis and meiosis. Okay, then cells can be stopped temporarily. The division can be stopped when they enter a stage called G0 phase or quiescent phase. So first we discuss about G1 phase. So during this time, organelles are reproducing, protein synthesis takes place, which is required for the growth and differentiation. So here this is the G1. Now after G1, usually we get S phase, that is synthetic phase. So this G1 is a gap phase, which is preparatory phase for the S phase. So here all the proteins and organelles or everything which are required for the S phase are synthesized here. Here the transcription is occurring, therefore the DNA will be uncoiled. So here it is an interphase, right? This is called as interphase. And in interphase, I told you chromatin will be freely distributed in the chromosome, freely distributed in the cytoplasm. So therefore, that is euchromatin, that is true chromatin, which is not coiled. Therefore, it is easily accessible for the proteins and enzymes which are involved in replication and transcription. Okay, and this stage is also variable, like each organism will have different amount of time. Then most cells that differentiate will do during this phase. So cells are arrested in the G1 phase, have capability of reproducing, and when sometimes without entering the S phase, they can go to G0 phase. And during G0 phase, they cannot synthesize, like examples, neurons of our brain. So they usually, after G1 phase, they enter into the G0 phase, and therefore they cannot replicate. So once if they get damaged, they cannot replicate. So therefore, so some can enter into some cells, they go back to the G0 phase, maybe due to the external factors, and in some cells they can again get back to the G1 also. So muscles are G0 phase. The next is S phase. S phase is called as synthetic phase. So it's the second stage of the cell cycle, where again the DNA uncoils, replication takes place, organelle replication occurs, and this ensures that whenever the daughter cell is formed, the genetic content should be transmitted. The next is G2 phase or gap 2 phase, where as the synthetic phase ends, now mitosis has to start. That means now the genetic material is replicated, but the cytokinesis has to take place. Cytokinesis means separation of cells. So during this time, the cell prepares for mitosis, making necessary proteins such as tubulin to construct the microtubules and spindle fibers, which are necessary for M phase. Few hours, 4 hours actually. During mitosis, the nucleus is replicated and the cytoplasm of the daughter cells. So here DNA is replicated in S phase. These phases are triggered by accumulation of cell signals. Okay. So this is the how controls of the cell cycle. So the control of the cell cycle depends on the signal in order to get functional. Okay, see here the chemical signals which are not phosphorylated at the top, but at the bottom they are activated. So here there are two types of enzymes called cyclins and kinases. So cyclins are the family of proteins that control the progression of cells through the cell cycle. And there are another enzymes called cyclin dependent kinases. So kinase is the enzyme which transfer the phosphate group. So these kinases, when they are dependent on the cyclins, they are called cyclin dependent kinases. Only with the cyclin, the CDKs will become active. Okay. So I told you kinase is an enzyme that transfer phosphate group from high energy donor, that is ATP, and this process is called phosphorylation. So we have cyclin D, E, A, B. So from G1 to S phase, cyclin D will check whether the process has completed properly. Then from S phase to G2 phase, cyclin E. Then G2 to mitosis, cyclin A will check. Okay. Then during mitosis, cyclin B plays an important role. That means they are like a checkpoints, which will check whether the process is completed properly or not. And certain kinases may have two forms, the active form and another inactive form. And kinases that are phosphorylated, usually they will be in active form. Okay. So these cyclins, they will activate these kinases. Okay. So here you can phosphorylated. Phosphorylated signal comes. This comes and binds to the CDK. And here S cyclin will come and bind. Okay. Then it will get activated. This is for M cyclin. So you have different types of cyclins which will activate the cyclin dependent kinases. Okay. So they will form a complex, cyclin and CDK complex, regulates the mitosis portion of the cell cycle. Okay. So M phase promoting factor, therefore it is called MPF. So during mitosis, like all these, DNA replication or cell division. So these cyclins will check at every stage whether the DNA replication is properly done or not. If it is not done, then it will stop the cell cycle there. It will repair the DNA and again it will continue. If at the end of the cell, if it is not properly divided or so, it can like signal for the apoptosis or programmed cell death. So this MPF promotes the entrance into the mitosis from G2 phase by phosphorylating the multiple proteins. So the steps that it follows are nuclear lamina gets depolarized, causing disassemble, and it will, which in turn causes the nuclear membrane to disassemble. So the membrane will disappear. And the H1, that is histone 1 proteins, comes and binds to DNA in the chromosomes, and the chromosomes condenses, and the cytoskeletal proteins will get involved to form cell division is performed if everything is done correctly. So once the CDK phosphorylation gives certain signals, then the cyclin will get destroyed. Cyclins will rise and fall, okay, based on the signal. So this is how it raises between M cyclin concentration will change during. So here you can see in G1 as G2, you don't see any activity of MPF or M cyclin. Only during M phase it is raised. So the term cyclin was Hunt, who discovered the cell cycle of sea urchins, and he said he named it after his hobby of cycling. So whatever he named it as a cyclin. Okay, and this, as this is a cell cycle, so the name itself, it is appropriate here. So the cyclins, so which will again fluctuate its levels during the cell cycle. So they received Nobel Prize for medicine in 2001. So different types of cyclins are there. So G1 CDK. Okay, at G1 phase, cyclin dependent kinase complex. So here cyclin D, which will allow the transition from G1 to S phase. Then G1S, this is the phase, and CDK. So here cyclin E, this binds to CDK at the end of the G1 and commits the cell to the DNA replication. Then in S phase, cyclin A, it checks whether the DNA replication is properly done and it is necessary for initiation of DNA replication. Then MCDK, where cyclin B. Okay, so this cyclin, this promotes the events of the mitosis. So how they control the cell cycle? The cell cycle has number of external and internal, and they will be checking the how the processes are taking place. So in first, that is G1S checkpoint, the primary determining factor for cell division to takes place. So this is the first step. So growth factors are affecting the cell cycle and cells are growing here. Once the R point is passed, now the DNA gets replicated. If the cell will get a signal to go ahead, then it will complete the cell division and cell cycle. If it is not getting the signal, then it should go back to the G0 phase. Okay, this is first checkpoint. Then second checkpoint is G2 checkpoint. Here, this ensures that DNA is replicated properly and there is no DNA damage. If they combine, they form maturation promoting factor or mitosis promoting factor, MPF. And then comes the next checkpoint, that is M spindle checkpoint, which will ensure that all chromosomes are attached to the spindle. So for mitosis, and the separation of chromatids is irreversible. So once the chromatids are replicated, they are bound together to a protein substance called cohesin. So cohesin is a protein where they both will bind. So another protein called separase. Okay, the separase will destroy this protein cohesin, but again it has two forms, active and inactive. So that means at every checkpoint, the checkpoints will look for whether the process has taken place properly or not. And if it happened only, then only the process will go further. If it is not properly done, then the cell cycle will stop. So sometimes, very rarely, even the cell, if it is not dividing properly also, it will somehow skips the checkpoints, and that kind of cells will become cancerous cells if there is any defect. So there are some external signals also, like nutrient and regulatory proteins. So external growth factors can regulate and determine the cell division in mammals. For example, when skin has some damage or wound, the platelets are released a substance called platelet-derived growth factor. So this will induce the fibroblast cells to start the cell division. But due to the external signals also, the cell cycle will start. And cell reproduction and healing of a wound. So this is a simple example. So we have many more conditions where first is homeostasis, where the bleeding is contained via blood clotting. First, bleeding is controlled. So that means the bleeding has to stop first. This is by blood clotting. Then inflammation. So bacteria and debris are removed and the damaged platelets release platelet-derived growth factors, and they will send some growth factors, so therefore they can reproduce and make new cells. Then proliferation takes place, so the fibroblasts reproduce, forming an extracellular matrix, and remodeling using some extra collagen and cells, the wound will be healed. So the same thing again, like how G1, so this is a G1 phase, how the here you can see the chromatin is dispersed here. Then in S phase, this will get replicated. Okay. Then mitosis takes place. Okay. So in the next class, we can discuss about different stages of mitosis and also cell signaling. Okay. So if you have any doubts, you can just ask. Is it clear? Okay, if you don't have any doubts, okay, let us stop the class here. So I have shared the PPTs in the group. Some PPTs I have shared. So I will share rest after the class. Okay. Thank you so much.